Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So, we were learning about cell biology, in which we have already discussed about cytoskeleton, the actin filaments, microtubules, and intermediate filaments that make up our cytoskeletal system. We discussed about each of them in detail. We also discussed about the different models of plasma membrane that were given by different researchers, which gave us idea about how the plasma membrane is made, what kind of molecules are there in the plasma membrane and what is the probable structure of plasma membrane. Today we'll be discussing about the composition of plasma membrane and the structure of plasma membrane in detail. So in last class we learned that the Singer-Nicolson model, the fluid mosaic model, is the most accepted one. It is the appropriate model which tells us about that a model that the plasma membrane is fluid, means it is a fluid of lipids. It is a sea of lipids in which proteins are embedded. Let me show you a schematic for that. So here you can see this is an electron micrograph of plasma membrane. So this plasma membrane is viewed here in a schematic in a cartoon form. So here you can see in the lipids, in the sea of lipids, membrane proteins have been embedded like icebergs. So this is the representation of fluid mosaic model, which was given by Singer and Nicolson. So the thickness of lipid bilayer is 5 nanometer. So the plasma membrane thickness is 5 nanometer, in which there are protein molecules inserted as well as cholesterol inserted. Okay, so let's learn about the composition of plasma membrane. What our plasma membrane is made up of. Okay, so plasma membrane has a major portion of lipids. Okay, it also has proteins and it also has carbohydrates. Carbohydrates, they are oligosaccharides that will be added on the lipid groups or the proteins, membrane proteins, the glycosylated proteins. So that's how carbohydrate is also a component of our plasma membrane. So the percentage of lipid and protein varies from different plasma membranes. For example, the plasma membrane of RBC will have different lipid to protein ratio as compared to the plasma membrane of neurons. So depending upon what kind of function that plasma membrane is going to serve, the protein to lipid ratio varies. Okay. For example, the plasma membrane of neurons, which are myelinated neurons, that they will have high lipid to protein ratio because lipids will be higher because basically they are going to only provide insulation, while the functional plasma membrane, which is selectively permeable, will have higher proteins compared to lipids because it is going to it is going to be metabolically active, so it has to do the trafficking of a lot of molecules, so it has to take in or give out certain molecules, and that is why the ratio of proteins to lipids will be higher because number of proteins should be high. But by convention, generally we can say that 50% mass of plasma membrane is made up of lipids, while 50% mass, rest of the 50% mass of plasma membrane is made up of proteins. Okay. Even though the ratio of lipids is higher, let's say in the RBC, 75% are lipid, and the rest of plasma membrane is 25% of plasma membrane is made up of proteins. This is the case of RBC plasma membrane, let's say. But lipids are smaller molecules, while proteins are bulky molecules. So they are going to contribute in the mass of the plasma membrane more, even though the percentage is less. So by convention, half of the mass of plasma membrane is made up of lipid, while the other half is made up of proteins. Okay. So let's learn about different types of lipids that are present in today's class. We'll only learn about the different types of lipids that are present in the plasma membrane. So the lipids that are present on the plasma membrane, they are categorized into three main types. Okay. So the first category and the most abundant one is phospholipids. Then plasma membrane also has glycolipids, and it also has sterols. Okay, the phospholipids, they are again categorized into two types: the phosphoglycerides and the sphingolipids. Okay, so let's learn about all of those different lipid molecules that are present in our plasma membrane in detail. So the very first one is phospholipid. Okay, so let me show you the molecular structure of phospholipid. So, so this is a phospholipid molecule. This is actually phosphatidylcholine. So it is a phospholipid. So here you can see it has, as discussed in last class, the lipid has a polar head group as well as a non-polar tail group. Okay, so this looks quite, this looks quite tricky to remember, but fret not, it is quite simple. Let's break it down into components. So all of the phospholipids, they have been categorized into two categories depending upon the backbone they have. So phosphoglycerides, they have glycerol as their backbone. On the contrary, sphingolipids have sphingosine as their backbone. So let's learn about those molecules first. Okay, so let's learn about the molecular structure of glycerol. So glycerol has three carbons. The glycerol has a three carbon chain, okay, on which each carbon has OH group. Okay, this is a molecule of glycerol. So on this glycerol, when you add three fatty acid carbon chains, so three hydrocarbon chains, three fatty acids. So what are fatty acids? So they should have acidic group. So how would a structure look like? It will have an R group with with a carboxylic group. So this is a representation. This is the formula for fatty acid. Okay. So three different types of fatty acid, when they react with glycerol molecule and they replace the hydrogen in the hydroxyl group one by one, that will give us the triglyceride. Okay, the simplest of all lipids. So let's write down. Let's note down the formula, molecular formula for triglyceride. Okay, so here we are on the oxygen on each OH group. We are removing hydrogen and we are adding the fatty acid group. So let me write it down like this. So let's say that this is Rn. So this R has n number of carbons. Okay, so R dash will have n minus 1 carbons, CH2, COOH. Okay, so this is the same fatty acid because R dash plus 1 is equal to R. Number of carbons in R dash plus 1 carbon is R. So both of them, they are the same fatty acids. Okay. So when we add this fatty acid on the glycerol backbone, it is going to form ester bond. Okay. It is going to form ester linkage with the glycerol backbone. So this oxygen from the glycerol, and this is how ester linkage looks like. Okay, so this is the carboxylic group, right? And this is the ketone group, right? I hope you remember. So carboxylic group has this structure. So when this oxygen, it interacts with certain molecule, so it forms a bond with certain molecule. This linkage is known as ester linkage. Okay, so this linkage is ester linkage, while oxygen bonding with two molecules, this linkage is known as ether linkage. So here this type of linkage is formed. So ester linkage is formed between the glycerol backbone and fatty acid chain. Okay, so it will have R1. Let's say here different type of fatty acid has been added, and here different type of fatty acid has been added. So this is a molecule of triglyceride with three different fatty acid chains which are forming ester linkage with the glycerol backbone. Okay, so this is a triglyceride. But when on this triglyceride a phosphate group is added, okay, when on this triglyceride a phosphate group is added, then we call it as the phospholipid. I mean, that is a intermediate step to form a phospholipid. That is not quite exactly phospholipid. Okay, let me draw the ester linkages R2 and R3. Okay, so here we are replacing this fatty acid chain with a phosphate group. Okay, so this is the phospholipid. This forms phospholipid, this molecule, when here OH group is added. So here a moiety having OH group is added. Let's say here choline could be added. Here we could add serine. Here we could add ethanolamine. Then that is going to make a phospholipid. Okay, I hope the structure of phospholipid is clear to you all. Let me redraw it. So how a phospholipid will look like? It will have a glycerol backbone. Okay, a glycerol backbone to which there will be two fatty acid chains. Okay, and on one carbon of the glycerol backbone, we will have phosphate group. I'm denoting it as P for our convenience. And on that phosphate group, there will be any OH containing moiety, any OH containing group like choline or serine or ethanolamine. Let's say here we are adding choline group. So this phospholipid will be known as phosphatidylcholine. If we add serine on that, it will, if we add ethanolamine over there, it will be called as phosphatidylethanolamine. Okay, so I hope the structure is clear to you all. Let's take a look at the schematic of the structure. So here, now you can see it has a glycerol backbone and fatty acid chains, to which two fatty acid chains are attached. To the third carbon of the glycerol backbone, there will be a phosphate group attached, and here this is the choline group which is attached to the phosphate group. That is why this is phosphatidylcholine. Okay, so there are other phospholipids such as phosphatidylethanolamine, phosphatidylserine, phosphatidylcholine, sphingomyelin, and sphingosine. We are going to discuss that later because we are separating them in a sphingolipid category, even though both of them are phospholipids. So let's just extend this map into, so lipids have phospholipids, glycolipids, and sterol. Three different types of lipids are present on plasma membrane, in which the phospholipids are categorized into two categories: the phosphoglycerides, which have glycerol as backbone. Okay, so we saw depending on which kind of OH moiety has been added to this phosphoglyceride, they are again of three different types: phosphatidylcholine, phosphatidylserine, and phosphatidylethanolamine. Okay, then the sphingolipids, instead of glycerol, they have sphingosine as their backbone. So let's have a look at the sphingosine molecule. So now you remember the glycerol molecule. It has three carbon chain to which Each carbon has OH group, right? So this is glycerol. Sphingosine molecule is no different from glycerol. Uh, in case of a sphingosine molecule, we can say that it is a it is a extended version. So it is going to have different it is going to have different groups compared to the glycerol. But if you remember the structure of glycerol, you can easily remember the structure of sphingosine molecule, because as the glycerol molecule have 3 OH. Okay, so the one OH group will be removed, and if you add NH2 group there, and if you add one fatty acid chain to the to one carbon, then this molecule is known as sphingosine. Okay, so what is a sphingosine molecule? On the glycerol group, when you remove one OH group, the OH group from middle carbon is removed, and there you add NH2 group, and you add a fatty acid chain to one carbon. That is what we call as the sphingosine. So this is the backbone for the phospholipids, and when this is the backbone for the for phospholipids, that phospholipid category is known as sphingolipids. Okay, the most abundant sphingolipid is known as sphingomycin. Sphingomycin. Okay, so I just made a mistake here. It's sphingosine and sphingolipids. Okay, so the most abundant sphingolipid is sphingomyelin. Okay, so on this, so let's have a look at how from the sphingosine sphingomyelin is formed. So this is sphingosine, right? OH. Here there is NH2, and here there is OH as well as a long fatty acid chain. So when on this NH2 group, when you remove one hydrogen and you add one fatty acid chain, so when you add a fatty acid chain on this NH2 group, here you have your sphingomyelin. Okay, so I hope the structure of sphingosine and sphingomyelin is clear to you all. So now it will be easier to remember how the structure is formed. Okay, so the fatty acid group is been added on the second second carbon. Oh yeah, I forgot to mention that on the third carbon there will be usual the phosphate group, okay, with ethanolamine OH group. That is our sphingomyelin. Okay, I just forgot to mention this part, that with the oxygen, there will be a phosphate group attached, the phosphate head group. Only then it will be a phospholipid, right? And with that, the OH group will be ethanolamine. Then we call it as the sphingosine. Okay, so let's have a look at different let's have a look at the different phospholipids. So these are the major types of phospholipids that are found in our plasma membrane. So phosphatidylethanolamine, this is a phospholipid, so it has a phosphate group. The ethanolamine group, it has a glycerol backbone and two fatty acid chains, right? The phosphatidylserine, it has glycerol backbone, phosphate group, the serine molecule, okay, the serine amino acid, and two fatty acid tails. Okay, then phosphatidylcholine, it has again phosphate group, a glycerol backbone to which two fatty acid chains are attached, and there will be choline moiety to the link to the phosphate group. This is sphingomyelin, where you have sphingosine as the backbone. So the sphingosine is the the NH. This is the sphingosine. This is sphingosine backbone. To the sphingosine backbone, there will be a phosphate moiety attached. So this is this is the OH moiety and the two fatty acid chains. So these are the structural, these are the structures of the different phospholipids that are found on our membrane. So then the second category of lipids that are found, they are known as glycolipids. Okay, so glyco means sugar and lipids. So glycolipids are the lipids over which the the oligosaccharides have been added. So when oligosaccharides they are added to the lipids, that is what we refer as the glycolipids. So in animal cells, glycolipids have sphingosine as their backbone. Okay, but in plant cells, glycolipids have glycerol as their backbone. So this is an important point to remember. Okay, and the most abundant glycolipid that is found in the animal cell, that is ganglioside. Okay, just remember the name. Ganglioside is the most abundant glycolipid that is found in our plasma membranes. Then the third category of lipids that are found on the plasma membranes are sterols. Okay, and the famous example, the most abundant sterols that are present in our plasma membrane and that act as the plasma membrane fluidity buffer, they are known as cholesterols. Okay, so let's have a look at the structure of cholesterol. So let me show you the structure of cholesterol. So this is cholesterol molecule. So a cholesterol molecule has, it has a hydrocarbon tail. It has, so instead of any backbone like glycerol or sphingosine, it has a plain steroid ring structure to which a polar head group is attached and a non-polar hydrocarbon tail is attached. So due to presence of a small OH as a polar head group, these cholesterol has a distinct property due to which it maintains the fluidity of membrane, and that is why it is also known as membrane fluidity buffer. We'll discuss about this in next lecture. For now, just remember that cholesterol is a sterol, the most abundant sterol present in our plasma membrane. It is a very important structure that is molecule that is going to provide the rigidity to the plasma membrane, and that is also going to control the fluidity of plasma membrane. So the cholesterol has a steroid ring, okay, and to the steroid ring there will be a polar head group attached and a non-polar hydrocarbon tail attached, and that is why this is also an amphipathic molecule. So with this, we are done with the composition of plasma membrane. So let's do a quick recap of the plasma membrane composition. So as of now, we are already we already know that the plasma membrane is made up of lipids. Plasma membrane is it is a basically lipid bilayer in which proteins are embedded. So it is a sea of lipids in which proteins are embedded just like icebergs. So the lipids, the different types of lipids that are found in plasma membrane are categorized depending upon their molecular structure. So there are in our plasma membrane, there are phospholipids, glycolipids, and sterols. The phospholipids are the most abundant type of lipids that are found in our membrane. So phospholipids, based on what kind of backbone they have been categorized into two types: phosphoglycerides and sphingolipids. Phosphoglycerides having glycerol as backbone and sphingolipids having sphingosine as the backbone. Okay, so we discussed about glycerol structure as well as sphingosine structure. The phosphoglycerides, that's why, are of three different types depending on the the polar head group that they have. Like on the phosphate, what kind of OH group has been added. So they, on the basis of that, they are of three different types: the phosphatidylcholine, phosphatidylserine, and phosphatidylethanolamine. If the choline is added on the phosphate group, it will be phosphatidylcholine, which is also abbreviated as PC, PS, and PE. Okay, so these are the three different type of phosphoglyceride. Then this in the sphingolipids, sphingomyelin is the most abundant sphingolipids that is present in our plasma membrane. So that is the worth remembering example, sphingomyelin. Then the glycolipids, they are the lipids on which the oligosaccharides chain has been added. So in the animal cells, sphingosine backbone is there in the glycolipids, while on the contrary, the plant cell have glycerol as the backbone for the glycolipids. The most abundant glycolipid is ganglioside. Okay, if question has been asked that what is most abundant lipids in our plasma membrane, then you'll answer phospholipids, but the most abundant glycolipid is ganglioside. Okay. Then the third type of lipids, they are sterols, in which the most abundant sterols are the cholesterol that are present in almost all types of plasma membrane in eukaryotes. Cholesterol is absent in the bacterial cell. Okay, from the bacterial cell, I remember one more point. So here you saw that the glycerol backbone, okay, the glycerol backbone and the fatty acid chains, they form the this ester linkage, right? So this ester linkage is formed only in the animal cell, in the eukaryotic cell. But on the contrary, the archaebacteria, they have ether linkage. So this is the ester linkage and this is ether linkage. So instead of ester linkage, the archaebacteria have ether bond between the glycerol and fatty acids. So the branched and they also have branched fatty acids. So we do not have branched fatty acids in our molecules. So here the fatty acids that we have are usually they are of two types. They could be saturated fatty acids or unsaturated fatty acid. The saturated fatty acids like the 16 carbon fatty acid, palmitic acid, and the unsaturated ones being oleic acid, linoleic acid. Okay, so the 16 carbon saturated fatty acid that we have is palmitic acid, then stearate. Okay, the unsaturated fatty acids that we have, they are oleic acid and linoleic acid. Oleic acid have only one double bond at the ninth carbon, while linoleic acid have two double bonds at the ninth and the 12th carbon. Okay, so we only have straight like we only have the straight chain fatty acid tails. So let me show you the schematic. Here you can see we have, I guess this is palmitic acid. If this is 16 carbon chain, then it will be palmitic acid. And if it is, so here only one single double bond is present. So this is oleic acid. So we only have straight chain fatty acids in our phospholipids. But archaebacteria, they have branched fatty acids, which are also known as phytol tails. Okay, so this is very important point that archaebacteria have ether linkage between glycerol and fatty acid, and they have phytol tails. So the one more difference between the archaebacteria and the eukaryotic plasma membrane is that the glycerol that we have is the D-glycerol, but they have L-glycerol. Okay, so this is also important point. Yeah, you can remember this point as well. So the animal cell and bacteria, the normal bacteria, they have ester bond and unbranched fatty acids, D-glycerol, but archaebacteria have ether bond. They have phytol tails, so they have branched fatty acid, which are also known as phytol tails, and L-glycerol. Okay, so this is the difference between archaebacteria, bacteria, and animal cells. Okay, so only archaebacteria, they have phytol tails. We have unbranched fatty acids. They have L-glycerol as backbone. We have D-glycerol as backbone. And they have the ether linkage between glycerol and fatty acids, and we have ester linkage. Okay, so with this, we are done with the composition of plasma membrane. In next lecture, we will learn about how the proteins, they show different types of movements in the lipid bilayer, and how the fluidity of the membrane is dependent on different factors such as temperature and composition of membrane. We'll discuss about the asymmetry of membrane. What are the functional benefits of asymmetry of membrane, and why is it functionally important to have asymmetric plasma membrane? So with this, I conclude the lecture. We'll also discuss about the different types of the how the different types of phospholipids they contribute to the functional asymmetry of the membrane. So till then, see you. Bye bye.